So I am Dr. Anahit Hovhannisyan. So I'll just tell a bit short who I am and what is my background so you guys know what to expect from me. So basically I have done my bachelor's and my master's thesis in Armenia, in Yerevan State University in the biophysics department in the biology faculty. And then I basically started working on my PhD project in the Biochemistry Institute, but then the things were not going really as I was planning them will be. So I got a bit angry, so I started to apply anywhere that I could. And then I wanted to stick on to the eye research because my master's thesis was on the eye, and we in the eye research area have this joke that if you once touch to the retina, you just fell in love with it and you just cannot do anything else, just study the retina. So that's what happened to me. And yeah, and I knew that I wanted to do research on the retina, and I knew that I wanted to do research on the retina diseases like degenerative diseases. So that way I basically got a position in Germany, in Tübingen, which is known as a neuroscientific center for the visual research. We have lots of labs, and they do from the molecular genetics until the physiology and programming and behavior monitoring, anything that you can imagine. I was joined to the lab where I was doing the research on the retina, but at the same time I was studying the other cells in the brain, the glia cells, which maybe one day I'll talk about them if I will be invited again, because they are very fascinating cells and they control lots of stuff that we don't know still yet how they are doing that. Anyway, so after I did my PhD, I got invited into to the UC Santa Cruz and the Center for Joint Project, where I was again working with the retina degenerations, but at this time I was working with the healthy retina and trying to find how the recovery process works with it. And then I end up moving to Fresno, where I am currently an adjunct professor there. Anyways, that's short about me and why I wanted to talk about that I have biophysics background, so I didn't have that much of a programming background or data science background when I went to Germany, and I haven't ever had any idea that I would deal with the data analysis as much as I was dealing. And you basically have to do that. Knowing the programming languages nowadays, it is not really specialty, it's just a language that you need to know. And there are lots of areas in the world. It can be astrophysics, it can be chemistry, it can be biology or neuroscience or whatever. You are using that tools to basically make analysis or investigate or develop models and do some other stuff. So today what I am going to try to do, I will introduce to the neuroinformatics, the idea what it is, which is kind of a different way to say computational neuroscience. When it have appeared and what kind of areas it is covering and what you can do if you are in the computer science field in this area. Yeah, so that's the outline of my talk. But before I start, I just want to know how many of you have been at my talk yesterday? Okay, so that's nice. Okay, that's good. So I wouldn't really concentrate. I kind of will concentrate, but not that much on the details, just to explain how the retina works. But so how many of you are physicists? How many of you are data scientists, programmers, IT person? Okay. Biologists, any? None? Okay. Neuroscientists? Okay. Not. But do you know where the brain is? Mm-hmm. You know that we have sensory systems that we are using, which use brain to function. Okay, so that's a really good starting point to have. So now what I want you to do, I just wanted to remind you, whoever have been yesterday at the talk and whoever haven't been, so just to have an idea that our brain is very common system that we use every day. And the cells are acting as an electrical circuit. So if you have, let's say, a microphone, and you want to speak, you need to have an amplifier. Yes, so your microphone will be your input device, which catches your voice. The amplifier would basically amplify the signal, and then you will have an output, which is the speakers that you will speak, and audience will basically receive the information that I am giving. The same kind of thing is happening with the neurons. So you have a neuron which has an input system. So this blue part that you see here, that's the input system. It's basically, instead of just having one input system like a microphone, neurons do have many of them. And our brain basically receives information from every side, and it combines, it analyzes, and it basically gives us output as an action. But in order to do that, you need to have a very small unit, which is the neuron, single cell, which does it. Then you basically would have the amplifier, which is the axonal part. And then you will have the output, the connections that I was talking about. And that would be just an example of the 3D reconstructed neuron, which is kind of the same picture that we see in a brain from the single cell. And that's just a small portion of the brain. And the blue spots that you see here, they are all different cell types. And the different colors that you see here, they are different cells. And as you can see, they are all having lines, which are the branches, the inputs that they are connecting. And you can see as well longer lines that are the basically axons and output, which are receiving the information. So anyways, because there are some people who have been there, so I don't want you guys to be bored, but what I want to present with this slide, very important calculation. So just imagine that we have 86 billion neurons in our brain, okay? And out of that 86 billion neurons, each of the neuron is having thousands of connections. Just multiply that and just imagine how many different iterations and connections are there in a brain. And at the same time, there are glia cells, which are basically the cells, administrating cells of the brain, which are underestimated and not very well investigated, but they are actually playing a very big role into the signal processing, into the neural survivor, and as well, they are some kind of amplifying the information that goes through the neurons. And then basically we do have as well different kind of profiles of the neurons. Like, for example, we have a person who stands in front of us and we see the appearance, yes? Which tall, short, Asian, African, Armenian. You just look at the person and you are saying, okay, this person looks like this. The functional part, let's say we have physicists, we have data scientists, we have business world people, so they are basically functionally different. Everybody has different function, yes, in life. And the genetics is basically our genetical background. We are Armenians, Germans are Germans, and that basically makes us different from each other. But they are all different. So we can have Armenians, but doesn't necessarily mean that all Armenians are programmers, yes? That all Armenians are very tall. And at the same time with the Germans. So basically you have these three components that you need to put together, and that's why people are so different, and every single person is individual. The same is with the neurons. And that's why we are saying that although we do have grouping of the neurons based on their morphology or physiology, but when you combine these three different profiles, you end up having a tremendous amount of different neurons that we are dealing with. Kind of individuals. So yeah, so we have very, very big amount of interactions. So to establish some baseline on the neuroinformatics, I just want to say that it is including data analysis, response prediction, image processing, modeling of neural systems, processes, prosthesis development. If you look at this, you wouldn't really guess that this is neuroscience. Yes, it kind of can have an image processing, which can be in the movies or, I don't know, animations and so on. You will still have an image processing to deal with. Data scientists, data analysis can be from the business world or physics world or biology world. So you can actually see that the neuroscience kind of with a data scientific point of view or informatical point of view, you just get a data that you are going to work with. It's just different. Input system is just different. Okay. So as I was talking about yesterday a bit faster because I wanted to share my research, I think it would be nicer for you guys to get more into how the data accumulation or the problems are from the data scientific point of view in each profiles that I was talking about, the physiological, morphological, and the molecular. So that's why we will concentrate on them. And the challenges that we have in every single field is the data accumulation, how we are accumulating data. There are tremendous amount of the softwares nowadays available, but depending on your research question, you need something special. And in order to do something special, you need to know how to modify whatever is existing or even write your own scripts or whatever in order to accumulate the data correctly. And I'll talk about it a bit more details when we get into different aspects. And then the data processing, which is because you are dealing with the living organisms, so it's not just data accumulation that you just get zeros and ones. You have lots of noise, background noise that you have to deal with. And the algorithms that currently are existing are pretty bad at catching real signals because the background noise can be much higher than the real signal. And in order to do that, you need to find correlations in the data sets. And the correlations can be very different depending what you are searching in your data. The data analysis is another thing where you can do lots of sophisticated stuff, looking into different angles to the data, which is very important as well when you are looking into your research question because you want to look from all the angles. The predictive analysis, which is currently when we have a prosthesis, for example, how we develop it. We basically have some neurons which have some functions, and we are like, okay, in order to get the hand to move, we need to have this neuron to connect to this one, and very simple interaction that has to take place between the neurons. And you are basically developing very simple system with a prediction, yes, that if something comes in here, how the neurons will act and what will be output. That's a predictive analysis, and currently the prosthesis development is in a very high need, and as well, for example, with the retina that I am working with, the retinal prosthesis are currently a very big deal, let's say like this, because you are having lots of different neurons and lots of different cells, lots of different interactions, and in order to predict what will happen, it's very, very hard to do it. And I'll talk about it a bit later. And of course the modeling. So you need to have a model systems, let's say the eye, artificial eye, to put into the person, and then person can see. And we don't have really very good models nowadays that you can actually use. All compared to the cochlear implants or, for example, the prosthesis for the hands and legs. So there's lots of stuff to do in this field, in this area. So now let's move to the morphology part and see how the image processing would work. So we have here a neuron. Whoever have been yesterday, you actually have seen this slide already, but I want to go through it. So basically that's the mouse, the animal model, because we are doing experiments mostly on the animals before we go to the humans, because we want to see that something works. And how you choose the animals depending on your research question that you are asking, because the same investigation you can do on, let's say, rabbits or pigs or mice or squirrels or rats, but depending which circuit you are investigating, you need to look how similar that part is the humans in order to actually have a closer answer to the question that you have. So here it's a mouse, because these rodents are very small and they are basically having a population much faster. And as well, there is lots of genetic background, like investigation of the genetic backgrounds of these animals, and we have lots of genetically modified animals. So in order to answer some medical questions, we can use these animals very easily. So let's say I'm looking into the retina. So you need to take the retina out. And I don't want you to be confused. The eye and retina are very much different things. So the eye is basically combination, all the lenses, all the muscles, everything that you can imagine that makes the focusing and not defocusing. But the retina is part of the brain that is inside of your eye, and it is a very small portion of the eye. And you can see that's basically the retina here, which is showing. So the retina sends the information to the brain, and it is central nervous system, part of the central nervous system. So in order to look at it, you have to isolate it. When you isolate it, you have to add some coloring depending on the different proteins and so on, and try to look into the retina under the microscope. And we have different type of microscopes, and I will talk about it later. Depending on the research question that you want to ask, if you want just to look to the overview, like this is very simple fluorescent microscope, you don't need even to have a confocal microscope to do it. The confocal microscope is basically some optics is involved in there, that you basically have a very narrow beam, so you can have very high resolution with your scans compared to just the regular microscope if you look under on the tissue. And then you basically, if you zoom in, you can see different cells and look into the processes, connections, all of that kind of stuff. So I have a video here. Hopefully this will play. And I'll talk about the video later. So
we're heading into the center of a mouse's brain, into the hippocampus where memories are formed. Looking up, you can see neurons projecting to the surface of the brain. This is the work of Carl Diceroff and his team at Stanford University. By making the entire brain transparent, they were able to image it using a light microscope. They call the new technique clarity. Existing techniques for studying the brain's wiring are often limited to looking at very small volumes of brain, or they don't allow you to label genes or chemicals of interest. The advantage of clarity is that you can label lots of molecules in whole brains. So how do you make a brain transparent? The thing that obscures the view is fat. Lipid layers surround each cell. To remove them without disrupting the cell structure, the team used a hydrogel to create a mesh to hold the rest of the components in place. Then they could clear away the fat. This is a mouse brain before and after. The brain is now transparent to light, but it's also permeable to molecules, which means scientists can add molecular markers to highlight specific features. In this 1 millimeter block of hippocampus, excitatory neurons are green, inhibitory neurons are red, and cells called astrocytes are blue. The technique works in human brains too. This is a chunk of the frontal lobe of a 7-year-old boy who had autism. It's possible to trace the path of a single nerve projection through a forest of other cells. When the team looked closely in one layer of the cortex, they noticed ladder-like patterns where neurons had connected back to themselves and to other neurons. Similar abnormal structures have been seen in animals with autism-like behaviours. Being able to analyse brain structures like this and match them up with molecular information could help neuroscientists uncover how changes to the brain underlie disease. And it's not just the brain. The researchers say the technique could be applied to any organ.
So I'll just stop it in here. What you have seen in this video, what I wanted you to see, first of all, that you can see how many different cell types and neurons are in a brain. And in order to actually see them, and it's a very hard question as well to ask, because the lipids, as you know, they are adding like non-transparency to the brain. So if you try to image cells which are deeper into the brain, you need to slice them. And when you slice, of course, you lose some information. And the researchers were trying to find ways to get rid of that lipids in order that they see only proteins, because this technique is basically you are eliminating all the lipids and you have a proteins which you can investigate. And the cell structure itself is basically a combination of the different proteins which are held by the lipids. So if you fix the tissue and then remove the lipids, you will have the exact positions of the proteins, and you can actually look much clearer. Like you can just take whole brain and image through it, and there will be no clarity issues with this technique. And this is very nice one if you are trying to look into the different connections of different cell types, or if you are trying even to make it like with the autistic brain. Yes, you can just have normal and disease brain and then try to address question what is the difference in here. But the problem with this technique is as well the data, because this is very tremendously big data sets that you are accumulating. And in order to look into the morphology of these cells, you need to basically do tracing or tracking. And as you can see, how many neurons are in there. And it's not hard just to go with the hand and do all of this. So you need to develop kind of image processing platform to make much more clear images in order that scientists can look at it and say, okay, these are the differences, and I can see the molecules and proteins are connecting to each other and so on. So this is one of the techniques that are used. It's not that very widely used, but because if you are trying to look into very individual problems, and this technique is not that much used because nowadays you want to combine physiology, histology, as I told, morphological part, and the genomic part. And when you want to have a physiology, you want to have a normal brain, functioning brain, alive brain, in order to see how neurons are talking to each other, because if you fix them to do histology, you cannot see that. So this is another video that I want to show you, and this is an electron microscope which will give you much more clear view into brain cells, because the other one was just giving you an overview. But in order to see the connections, you need to have much higher resolution, much closer look to that.
Trying to introduce the study of the brain to a bunch of students. I said, if understanding everything you need to know about the brain is a mile, I asked the students, how far have we walked in this mile? I got answers, 3/4 of a mile, half a mile, someone said 1/4 mile, and I said, I think about 3 inches. This is unconventional science. There's just this huge gap in our knowledge about how brains work, largely because we have no idea what they actually are made up of at the finest level. When it comes to the nervous system, there are a large number of diseases where the only real sign that there's something wrong is the outward manifestation of the disease. A person is acting crazy, or they don't seem to learn very well, or their movements are disordered in some way. But if you look at their brain, most of the techniques we have, there's nothing to see. You've got to see the wires. You just have to see them, and you have to see where they come from, where they go, what they connect with, and be able to map that out in enough detail that ultimately you will be able to render the information that goes into a brain to see the wiring diagram at the resolution of every single synaptic vesicle in every synapse. Enough resolution, we think, to see everything in a wiring diagram. I'm a lab head of a group of people who work together. It's kind of a shifting group of people, but it's difficult work, and it requires many kinds of experts. So there are lots of people working together on this project. What we're trying to understand is how cells in the brain communicate with each other. I have to look at the entire pipeline from the beginning, where we start with the mouse, make a sample, process the tissue, cut sections, and then ultimately begin imaging them. A slice is like you're cutting a loaf of bread, except you're trying to slice very thin pieces of tissue. So for a millimeter depth, you get about 33,000 sections. Each section of the brain is next to the section that was sliced just before it, and the slice right after it is the next frame. And if you play these frames in sequence, you see the brain not over time, but over space, as if you're looking deeper and deeper into the brain. The big objects you see that appear and then slowly go out of appearance, those are nerve cells that are being cut through. And coming off of these cells are these large, light-colored objects, which are the dendrites. Every little object in here is a little wire. To keep track of these cells from one section to another, what you'd like to do is kind of color them in. So every one of these colors have no meaning other than to keep track of each object from section to section. What this allows us to do is to generate a wiring diagram based on this. We now know everything about these wires here, and from that we can generate all the connections of every cell in that area. Information forces you into this uncomfortable position where you have to kind of say, okay, I don't get it, but I know that the real world is more complicated than the way I'm thinking about it. I feel it's a very long road, and we've just started. That's my view.
So what you have seen here is the electron microscopy technique that is used. An electron microscopy technique is used, I think it started from the 19th century, or actually 20th century, where people found the technique that by using just an electrons, you can see where are the membranes or the cells and everything. So all the gray coloring that you see, that's actually the image that you see there. And what the challenge in here is for the data scientists or image processing people is to find the correct pieces of the puzzle, because you basically just imagine, he told like 1 millimeter of tissue, see how big is our brain. Just from 1 millimeter tissue, you get thousands of slices. And the cell size, the average cell body size is only 10 micrometers. Just imagine how many cells you will have in just one section. And in order to see how the reconstruction works, like you have seen this colorful images and branches and so on. Basically, you have to go through all the sections, find correct pieces, align them together, and just get the cell. And of course, then you have different cells, and then you look into the connections. That's the next step of it. And this is lots of work. Like I was working with the bipolar cells, and in Germany, one of the groups was having like 20 different students for a couple of years just to work on reconstruction or one individual cell. So it's very, very hard process, and you need to actually write the scripts from the scratch sometimes, or have some things that are already written, develop them and change them in order to actually do the tracking correctly and reconstruct the cells correctly. So this is just only about the imaging part of it. So neuroimaging data accumulation is requiring lots of time, as you can see. It requires higher resolution because that's a big issue for us. The clear technique that you have seen, the transparent brain, of course it's cool. You can see the cells just right away. You just put the specific stainings, and then specific cells are stained with the different colors, and you are like, okay, that's interesting. But when you want to see the connections and you want to see how different are the cells and to dissect more of the information or the diseases and go into the more to the functional direction with the morphology, you need to deal with the higher resolution. And that's still, we are still on the edge of the technical development. We cannot really get there. Data size is another issue. We are dealing with lots of data, so which means that nowadays the programs are not really that easy to use because you have to use lots of memory and lots of high resolution, like a speed. You have to have gaming computers and so on. And I had very hard time as well to do my analysis because you had to wait quite long time until your data is reloaded and you do some changes and then you look at it. So you have to actually change your data into the much more smaller format depending which information you want to dissect from your data sets. And of course the methods are another challenges that we have. The fluorescence microscope, which was used on the clear brain. The confocal microscope, which as well was used in the clear brain, but you just can go much more zoomer, much more closer look. You can have much more closer look to the cells. 3-photon microscopy, which is very interesting. The 3-photon microscopy is pretty new thing, and I was lucky to work one of the innovators at that field in Santa Cruz University. So the idea is that in order to get deeper into the tissue, you remember I was talking about lipids that are making much harder to go deeper into the tissue. In order to do that, you need to use not 1 photon, which is the, let's say, confocal microscope is only 1 photon. You are using 2 photons to go deeper into the tissue and try to look what's happening in there, or 3 photons. And this is used especially for the lifetime imaging when you have an animal under the microscope and you want to go deep into the tissue and try to look into the deeper sections of the brain. Like hippocampus is pretty deep, so you need to go deeper in there to look at it. And another thing is that the analysis. So image processing, the colocalization, like which is shown down here. So you can see 2 different colors. Yes, you have the red and then you have a green, which are, you can just imagine that the cell ends different proteins, the input and the output. That's going to be your input and that's going to be your output, and you want to see if these proteins are actually coming together. Wherever you see yellow or orangish color, that means that there is a possible functional connection between these 2 cells. So that's the colocalization technique, which again you need to have much higher resolution data in order to do this. Also process tracking, when you are trying to find a single cell and track the processes to kind of see where they go and how they connect to each other. And the last thing is basically 3D reconstruction, the video that we have seen previously, which that's each color is basically 1 individual cell. And the tracking, as I have mentioned, it's very hard to do and requires lots of time. So we are done with the morphological component. If you have any questions, I can answer to them. Okay, no questions. Okay. I usually tell if people don't have questions, that means that I'm not giving good information because it's not clear. I hope everything is clear to you. Okay, so now let's move to the physiological part. And here I have a very short animation which shows how the signal travels through the neuron. And you can see that's the cell body part, that's the input. And this is the amplifier part. And the output you will see in the background. Okay, let's see. So basically, signal gets into the cell body, and it travels backwards into the signal transduction path, and then to the output part. And it's basically electrical signal, literally. It's an electrical signal because the electrons are traveling through the axons, and the information is like a wiring in the light, switching on or off the light. That's how the neurons do work. Here I have another cool video. I hope it will be cool for you because it is cool for me. If you remember, I was talking about 2-photon and 3-photon imagings. That's the 2-photon imaging, going deep into the brain and trying to see how the neurons are firing. Firing means if you get an information, the neurons send too much electrical signals. And whenever you see that signals, you will see the brighter green color. And the green that you would see, it basically would be the cell body, so the part that receives input or information, because that's where most of the electrons are located. So let's see if it will play. Hopefully it will. I don't know, the contrast is not that good. But now, can we switch off the light? Maybe dim it a bit. Sistem hanım. Okay. What's up? No. Սխալ արեցի։ Ես որ սխմեմ կանջատվի։ Չգիտեմ, okay, մի քիչ ավելի մութ ասա։ Հիմա փորձեմ մի հատ էլ վանեմ, կարող է եւ օգնի։ Իմաստն enough, so the idea is basically all these round things that you see, you would see the more greenish part, and whenever there is a signal coming through, you will see them green. So let's say if I have a retina and I'm showing like black and white screen to my eyes, the brain will receive that information. It will receive, okay, it was shown black and white. As soon as the cell sees the black, it will fire, so it will be green. As soon as it sees white, it will fire again. You will see again green. So ideally, our visual system, I'm just bringing example, our visual system is able to see the changes. So whenever there is any change, you will be able to see. If there is no change, your retina doesn't really percept that. So idea of this that I wanted to show you, first, this is for the physicists. If there are some physicists, I think there was one, at least in the beginning when I asked. So you are going to develop the very good optical systems in order to go deeper into the brain to catch the signals. For biologists, you are trying to have the animal in a good stage that they can actually respond to the information. For the data scientists, they get this data and they have to actually take out the information and tell in which time point the brain was active, when it was active, how long it was active, and how deep it was when it was active. And yeah, so basically, as you have seen in the electron microscopy video, there are lots of different area scientists that are coming together and working together. And the neuroscience is super interdisciplinary field, so anyone can work in here. So another thing that I'm going to make it even more complicated. So just imagine, that's the neuron, yes, that I was talking about. So that's the input part, that's the amplifying part, and that's the output part. What you see here, these blue things, these are the responses. And depending where you are doing the recordings means that basically you have an electrode which senses the changes. Whenever you have whatever simulation, like the retina sees the black screen, you will have the information, electrons will start to travel through the cell, an electrode will detect that. Depending where the electrode touches to the cell, your recording will be different. And you can use this information even to reconstruct a physiological profile of the cell, which means like, okay, you have only electrical signals, and by the shape of the electrical signals, you can tell where exactly the cell is located. Just imagine then you have the tissue afterwards fixed, you stain and you see the morphology, and then you see on top of that physiology. And you can try to see actually where there were active connections, where the cell was actually receiving information, because sometimes the view can be different. You will have lots of processes, but none of them can be active, or you can have very few of them that active. With this technique, you can actually see the active and fake processes, if I can put it this way. And the shape is very important as well for the data analysis purposes. This is just a setup. Oh, what that means? Oh, that's it's okay, we will make it. Okay, so that's the physiological profile. Again, from yesterday's talk. I just want you to have an idea how the recordings are done. Again, we are taking the retina out, okay, but this is alive. It's not fixed. So the connections are there, the proteins are moving, electrons are moving. And what is interesting, you can actually record from the single retina for hours, like even for 12 hours, you can do a recordings if you have normal physiological situation that biologists have to take care of. You will be able to record lots of signals from these cells. Then there are these chambers that you are putting your retina on flat, and they are electrodes, which are shown here. Okay? These electrodes touch to the cells and they catch the signals. And then you are showing different light stimulus, like as I told, black or white screen, or they can be natural scenery or moving gratings or whatever you want to investigate. And then, of course, you will get your responses, which look like this. So which means your electrodes were able to catch different neurons, and each neuron, each color represents one single different type of neuron. So you can see as well activation points of the different neurons. And the picture that I have shown in the morphological part, actually it's the same tissue that was taken. So you basically have the physiological data, and then you fix, and then you look into the cells. The physiological part has even more complications than the imaging part, and that's the challenges that I am listing here. So the cell classification, I'll talk about it, about more details next. Neural response analysis, like the stimulations, how to present them, because you need to make correct stimulations as well in order that the retina sees what you want it to see. And then data processing, afterwards you have the data that you need to deal with, and the population coding to see how different neurons connect to each other and how they code the information. So just to have a closer idea, so you can see this grayish part is basically the retina on top of the electrodes. The all black dots are the electrodes that are receiving the signals from the retina. That's just a closer look. And what you see here, you see lots of like bluish lines. So that's going to be your noise, which means that electrical noise, because when you do recordings, you will get always electrical noise. And the sharp things that you see, these are responses that we call spikes. Okay, and when I have shown previously the whole neuron, that in different parts of the neuron you do have different shapes, that's what it is. So depending where you are recording, the spikes will be wider or narrower, shorter or longer. So this, what I want you to see here is again the numbers, okay? So you have recordings that you are performing only from 1 millimeter of the tissue, and retina is actually 4 millimeter in diameter in the mice. So the humans is bigger. Then you have about 20,000 electrodes that you are receiving recordings. And then you are dealing with 80 gigabytes of unprocessed raw data. And then you have, for 1 hour data recording, 1 terabyte size of the file. So which means you have lots of data points, and as I have told, the cell doesn't really have only the input part, it has as well output, and it's longer, so which means one electrode, a couple of electrodes would receive information for the same neuron, so you have to as well kind of filter out that information in order to see different neurons. So that's basically what I was trying to say. So you have a neuron, which is shown in gray, and you have different electrodes that will catch the information. And the shape of the response, the green one, is different because depending from where the electrode is catching the signal size and shape is different. But the timing would be the same. So that's how you would know that it comes from the same cell. And that's the data that you receive after high-pass filtering, which means you are removing the extra noise, and you basically see that whenever you showed, let's say, the black screen, you have a response. Then whenever you show this white screen, you have a response. And then basically, if you are filtering out with a high-pass filter, you just get only the responses. All of these lines are basically responses from the single neuron to the very, very simple stimulation. How many of you are familiar with the PCI, principal component analysis? Linear analysis of the data. Okay, that's one of the things that we are using into our data analyzing and the spike sorting algorithms in a neuroscience. So again, that's the neuron, and depending where you are recording, the shape of the response is different. And in order to deal with the data classification, you need to take into consideration different cell types. And in order to know different cell types, you have like a timing of the responses. You have how fast they are responding to the input. and different things. So with the PCI analysis, like here we have an amplitude, and you can see when you use an amplitude, two different neurons are aligned with each other, which means that the signal that electrode was catching, the amplitude was the same. So you cannot really with this tell that you have two different neurons. But when you look from the shape point of view, you see that they have different shapes. So which means that you can actually separate two different cells from each other. And whenever you have this type of data, it's still not 100% clear. You have still to go manually and do corrections. Even if you try to use the best algorithms, it's still not really clear we don't know how to make it better. So we are trying to work on that. So for the classification challenges, as I told you, it's the scalability. So we are dealing with lots of electrodes, with lots of data, with long recordings. Data file is super big, which is basically scalability, speed, and performance, the solutions that we need to kind of search for, that we actually have the proper analysis done. And the next thing that the challenging, you need to know as well what are happening in the field. Like there are tons of new programs coming out every year. So you need to know how they are working and what are the difference between the other one and what is your research question and what you actually want that they don't have it. Because sometimes you try to find an answer, but someone else have found it already. So you don't need to use or lose your time to come up with that. Yeah, spike sorting is reproducibility is very, very, very hard to do, and we still are doing it manually, which means that you still go and separate the clusters and try to see the differences and see if it did do very nicely. So one of the solutions that we came up with, it's the deep learning method, when you kind of try to address different parameters and basically your programming algorithm is kind of detecting the pattern of the responses. Like, as I told, the shape of the responses can be different, but it can come from the same cell. And the timing can be one of the things that plays a role. That the time that took the response was the same, but the shape was different, so it's one single cell. And the next thing is to do unsupervised quality matrix modeling, and the accuracy and reliability of the algorithms that are developed and used. The population coding, I'll try to, response coding, I'll try to go faster. So that's what I was trying to tell in the beginning. When you are learning how neurons do function and do work, you are trying to develop a models or predict. If I show these responses, neuron will respond this way, and that's the output that I will have. Do I want that? If I don't want, what I have to change in the input that I get the correct output. And that's basically one of the simplest versions of the artificial neural networks. When you have the input, you have the connections, which is the hidden part of the computation that we have, and the output. The problem with these networks is that they are, the size of the cells are different, the numbers of the connections are different, locations of the cells are different. Artificial neurons cannot really cover all of this. The speed of the information is as well varying because we have neural systems that they can change. With this kind of systems, you have to have fixed speed of the signal transduction. Power consumption, which is how much energy you use. When you have an artificial neurons, you have to have a power of the computer power to actually deal with lots of data. But our brain doesn't, it deals with the food, yes, that's the energy that we are consuming. So we can do much more processing ourselves than artificial system can do. The signals, how they are transducting, and the learning, because neurons in the real world, they learn. The neurons in the artificial world, they don't learn. Which means that if there are some experiences, they will change their response pattern or they will rewire. And that's what is very important that the artificial neurons don't have. So the challenges will be in the artificial brains. They are very, very behind to the neural ones, which is the brain development and the cell changes that are appearing. Then we have the brain can easily adapt to the stimulations and change their response patterns. and the brain dynamics, because the brains are very dynamic. And as I told, if you learn something, they will rewire, they will change their connections, but the artificial brain cannot do that. So in order to deal with this, we have to implement lots of machine learning techniques to deal with the lots of data, first of all. And secondly, we need to accumulate lots of data. And lots of data accumulation means that you need to think about the correct stimulus. Anything that in the nature will happen, in a visual system, you have to cover everything, because each neuron responds differently and your predictions might never be the same. So that's why you need to have real data recordings, develop your prediction algorithms that when you are developing the artificial, let's say eye, you can implement all of that and kind of cover all the situations existing there. And we need to have more precise algorithms and analyses developed. The genetical part I'm not going to cover, but as I told, that's another component of it, just to combine and to do, and bioinformaticians are taking care of that. And that's basically all of the three things together. I think most of you might be interested to this part, because here I have written down the programming platforms that are used to address the morphological and physiological problems. I don't know if you guys recognize any of these platforms, but ideally neuroscientists do have some big communities which are developing these platforms, or sometimes you can even go by yourself and do changes in the code in order to kind of fit to your needs. But so I know that lots of data scientists do use Python, so nowadays we are moving to the Python as well in the neuroscience field, because it kind of changes the data size. It is making it smaller, so you don't really need to have very high computer power in order to make the processing of the data. And another thing that maybe can be interesting for you guys, I have tried to think of anything that the neuroinformatics would cover, like computational neuroscience, where you use the mathematics and computers to basically make the models or develop brain models. Neuroinformatics, which combines informatics and the neuroscience together to kind of deal with lots of data and do lots of prediction analysis. Data scientists will deal with much better data analysis methods. The app development is very important as well, because lots of neuroscientists doesn't have programming background and they just want to have an app to go. Like, let's say if I do recording, I want to use this app and do my image processing. I want just to do the tracking, and I want to click to the button and it does the tracking. I give the parameters and it does it. And another thing, bioinformatics, as I told, for example, gene expression is done. Lots of the bioinformaticians are involved in it. Optical physics, developing optical systems to make the imaging much better. The animators can actually do very nice animations of how, that for the educational purposes, how different brain areas and regions do work based on the data that we already have. And the engineers which can work on developing the prostheses. So all of different specialties work together to basically make the neuroscience good. So to conclude, I want to tell that neuroscientists do really need help. They need help to develop new technologies. They need help to find better ways to deal with a large amount of data and to analyze them properly. Then we do as well need strong interdisciplinary connections, because the neuroscience is super interdisciplinary and it connects almost everything. Even psychologists or philosophers are doing neuroscience. So areas interest would be computer visual algorithm, motor learning, generalization, pattern recognition and prediction analysis, neural imaging, image processing, neural network analysis and neural modeling, and the artificial intelligence and predictive analysis and the clinical trials are in there to actually make if you do use the machine learning or artificial intelligence, you can speed up data analysis, accumulation, modeling process, and that will help really a lot to the clinical trials as well. That was it. So that concludes my talk. I don't know if